embryonic development, all of that. When I see you on Thursday, we'll wrap that up and quick mention about genetics. Okay, so most of the genetics content, again, you have covered in lab. It's only things that you have not covered in lab that I will be emphasizing. So we should be able to complete everything related to this unit this week. You are taking your last lab exam sometime this week, right? Thursday, Wednesday and Thursday. Okay. Okay. So next week you will take the last lecture exam. This is unit 4. We are following final exam schedule as set by CTC. So for classes that meet on Tuesdays and Thursdays at 9 a.m., our exam time is basically Tuesday. Okay, so Tuesday at 9 o'clock. So next week, Tuesday, 9 o'clock, you will be taking your last unit exam. Now, even though it's the final exam week, this course does not, we don't do a final exam for this particular course, A and B in general. So you're only taking a unit 4 exam. So this is including pH, electrolyte, water balance, that chapter, which I think is 26, and then female and male reproductive systems, what we need to cover today, which is fertilization, fetal development, all of that, and then a little bit on genetics. So those are the chapters that you will be tested on next week, okay? There's not a whole lot of you, but I think whoever's already on my list to take a makeup exam because you missed an exam sometime during the semester, and you have a valid medical reason for that. If it's an excused absence, I will be contacting you. So I won't be contacting everybody. If you're on my list and we've already talked about it and communicated about it, sometime previously during the semester, I will be sending out a Blackboard message only to you. So if you don't receive a message, it's all fine. Okay. This is just a makeup exam, which will also need to be scheduled sometime next week. So let's see. So this week, you should be wrapping up everything related to lab. As I receive exam scores, lab activity, final scores, I don't know what else. Anything extra credit? Yeah. Okay, genetics is extra credit, right? Okay, so as Ms. Gosnell completes all of that, she'll be sending all of those scores to me, and I will be updating my Blackboard gradebook columns related to lab. By the end of the week, everything related to lab should be wrapped up, okay? You also have your last block of homework assignments and IP activities due, I think, this Friday. So this weekend, I'll be updating all of that, right? So as we go into next week, the only column that should be missing anything is the unit lecture exam 4, okay? And we should be able to wrap up all of that. And I'll give you more information as we go through the next week or so, right? Keep looking for Blackboard announcements. As I make updates, I will post an announcement asking you to go look at the scores, making sure that there's no mistakes or discrepancies. All right. So let's go ahead and finish this up. So I actually did cover quite a bit about the ovarian cycle, but I want to review it with the slides so that we're tracking just fine. So we talked about all the changes occurring within the ovary. This is how the follicles develop, right? The whole ovulation that occurs mid-cycle. And then after ovulation, what gets transformed off the vesicular follicle is called the corpus luteum. So all those changes that are happening is what we call the ovarian cycle. So your textbook cycle describes the ovarian length or duration as 28 days. So of course, there could be variability there. So if you consider a 28-day cycle, then basically all the events prior to ovulation is what I call the follicular phase. All the events after ovulation, right, till the end of that ovarian cycle is called the luteal phase, right? So let's start mapping out these things as we add information. Okay, what am I looking for? So as we cover this information, I want to review a few things before I go ahead. I also want to map out simultaneously all the changes that are occurring within the uterus, right? So the ovarian cycle and the uterine cycle, and they kind of go hand in hand, and we need to be tracking at, before ovulation, after ovulation, all the changes that are occurring, not only in the ovary, but also in the uterus. I'm going to zoom in here. Are we tracking? Okay. So when you're looking at the ovarian cycle, which is a 28-day time period, for example, right? Mid-cycle, day 14 is when ovulation is occurring. So all these time, this entire duration prior to ovulation is called the follicular phase. And everything after ovulation is called the luteal phase. So we did talk about a few things related to this. So let's map this out. Okay, so we have the HPG axis that regulates all of these different, this is the hormone. Oh, sorry, yes, we're good. Yes, I had difficulty with it, but I'm good. Okay, the HPG axis is the hormonal control of this entire ovarian cycle. But of course, I'm going to also show you how this, well, mostly controlling the ovarian cycle, but I also want to tie in the uterine cycle next. So H stands for hypothalamus. So the hypothalamus is going to release gonadotropin releasing hormone, and that's going to target the anterior pituitary, right? Prompting the release of two different hormones here, follicle stimulating hormone and luteinizing hormone, right? And then these two hormones from the pituitary will then target the gonad. In this case, it was the same axis that we talked about for spermatogenesis. The gonad there was the testes, right? It's going to target the cystinocytes and the interstitial endocrine cells. Here, we're targeting the ovary, and I'm going to explain to you what's going to happen. But basically, we are promoting oogenesis, right? The whole process of oogenesis, which is starting from a stem cell, going through meiosis one, where you end up with what? Like a secondary oocyte and the first polar body. And then ultimately, if sperm penetrates that secondary oocyte, then forming the ovum and all of that. So this, but really starting to produce hormones such as estrogen and progesterone. Okay, so when it comes to the ovarian cycle, so I'm going to add another time frame here. So let's go with like day 5. What? Like we'll keep adding information as we go through the slides. The follicular phase is called that because you're seeing changes in the follicles as they develop and mature. So a follicle basically consists of the center, which is the oocyte. The oocyte contains the chromosomes. So we start off with a diploid primary oocyte with 2n46 chromosomes, right? And then that is wrapped around by a single layer of cells called the follicular cells. As this matures, it becomes so primordial to primary follicle, and then it becomes something called a secondary follicle. The secondary follicle is where you still have the primary oocyte in the center, right? But instead of one layer of cells protecting it, now you have multiple layers of granulosa cells. That's the difference there, right? And then that secondary follicle then becomes a tertiary follicle, which goes by a few different names, right? Tertiary, antral, vesicular, Graafian, all the same, right? So the most mature version of that follicle is the vesicular follicle. This is where you still have the primary oocyte in the center, right? And this was, if you recall, arrested in prophase 1. And then what happens around it is where those granulosa cells kind of restructure themselves, where you now have a fluid-filled cavity called the antrum, and then the primary oocyte gets kind of isolated from the antrum. And I'll tell you about the different layers that are formed. I'll use the slides for this. This whole process of going from primordial to primary to secondary to tertiary is happening here during the follicular phase, right? But to guide this process, during these initial stages here, during these first few days, right? This is when GnRH is released by the hypothalamus, which allows for follicle stimulating hormone and luteinizing hormone to increase, right? So the follicle stimulating hormone is what's going to target those primary follicles, allowing it to mature into secondary and so on and so forth. But as the maturation continues past day 5, right? Because around day 4 or day 5, GnRH, I'm going to shut this down again, where you do not have FSH and LH being released for the remainder of this period, just the first few days. This is important for selection of the follicles, and it's important to get the process started, where the primary follicles are starting to develop into secondary and so on and so forth. But after that, I do not want follicle stimulating hormone. I don't want luteinizing hormone, because what that's going to do is it's going to continue to recruit more and more follicles during this ovarian cycle. We're done recruiting, okay? And it's not just one follicle that's recruited. We talked about this. It's more like 5 to 8 something follicles that are recruited. Depends on each person. It's slightly different, right? Once those follicles are recruited, we don't need FSH and LH. That's why it gets downregulated. We're going to shut down this pathway, right? So about day 5 or so, you don't have FSH and LH because we're shutting that down. But as the follicles start developing, where you go primary follicle to secondary follicle to tertiary follicle, as that occurs, as these granulosa cells are developing, right, that's going to start increasing estrogen levels. Estrogen is what's going to keep this process going for the remainder of that time frame. Does that make sense to you? Yeah, okay. So let's add a little bit of information here. So it's the follicular phase. Okay, we don't all have a 28-day ovarian cycle, right? So some of us have a shorter cycle, some of us have a longer cycle, right? So the variation in the length of this ovarian cycle is here during the follicular phase. So it depends on how fast that estrogen builds up. It depends on how quickly these follicles develop to get to that vesicular follicle stage, right? Which is that mature follicle. Now also remember, let's say 5 follicles start this process, right? And they all start developing primary to secondary, they start maturing into that antral follicle. Not all of them are going to mature at the same rate or the same speed. So typically what happens is as you're approaching day 14, around day 13, you only have one dominant follicle, even though 5 or 8 or whatever started the process, that particular month, that ovarian cycle, not every follicle is going to be dominant. Typically about one follicle is dominant, right? But when we get to that stage, this is where we have to prepare for ovulation, okay? Does that make sense to you? So far, so good. The follicular phase is the part that varies because the time that it takes for that maturation process for the follicles to develop can vary. It doesn't have to be 14 days. It could be shorter. It could be longer. But here's the deal. Once ovulation occurs, this phase, the luteal phase, is constant. It's 14 days. Why? Because a little later on here, we said we're looking for HCG levels, right? All of this is quite well defined in terms of the duration. This is constant, but the follicular phase is where the variation could be. All right, so let's keep talking. It's just showing you what all is happening during the follicular phase, how those follicles are developing. Let's look at this schematic. So if you're looking at a cross section of the ovary, which you have plenty of slides set up for you in lab, take a look at it. If you look at the cortex region on the outside, you should see tiny, tiny, tiny little follicles. Those are mostly primordial follicles. That's what we females are born with, right? I mean, so you got these primordial follicles. Remember, that's the entire package. So what you typically expect there is a center called the primary oocyte with what? 46 chromosomes, right? It's diploid and it's arrested in prophase 1. That's what you got going on within those primordial follicles. What's protecting that primary oocyte? Just a single layer of follicular cells, right? So then in response to the FSH and LH, okay, so let's say 5 or 6, 7, 8, whatever of these follicles, they're going to become primary follicles. So this is where, okay, it's kind of generic. Really, this process of going from primordial to primary follicle takes a little longer. I'll come back to that point. But what's the difference between the primordial and the primary follicle? Nothing really, just the size, just a little larger, right? So this, you still have the primary oocyte right there in the center, still diploid. Remember, we just started meiosis one. We're rested in prophase one. We haven't yet completed meiosis one. And you have a single layer of follicular cells protecting that primary oocyte, correct? So the primary follicle is responding to FSH, correct? And it's going to then mature, become a little larger, a little bit more differentiated, becoming a secondary follicle. So what do you have in the center? No change. Primary oocyte, still arrested in prophase one, still diploid, correct? Except you have multiple layers of cells here. We call these granulosa cells, right? Now, these are the follicles. As they're maturing, they're going to start producing estrogen, right? So estrogen levels are going to keep increasing, and that's going to allow further maturation of this secondary follicle into a very early vesicular follicle. This is where you're going to start forming little cavities there filled with fluid. All these cavities are going to kind of combine together, forming one large fluid-filled cavity called the antrum, correct? So this formation of the antrum, fluid-filled cavity, basically isolates that primary oocyte, right, from the rest of this overall follicle. So I have my primary oocyte here. Again, still arrested in prophase one, still diploid, but now we're going to call this, all right, so we've got the primary oocyte. You have a layer that's kind of that grayish layer surrounding it. That's kind of a gelatinous layer called the zona pellucida. And then you have a few layers of these granulosa cells that surround the zona pellucida. We call this the corona radiata, okay? So really on in the center, what do you have? Isolated in the center, you have the primary oocyte, arrested in prophase one, plus the zona pellucida and those few layers of the corona radiata. Does that make sense? Yes, ma'am. Yes, exactly. So it's all primordial follicles at the time of birth, throughout childhood, right, till you hit adolescence, till you hit puberty. And then as this HPG axis starts to become active again, right, then in response to the GnRH, in response to the FSH and LH, we're going to start the whole, like, let's get started with the ovarian cycles. Nothing is rhythmic, nothing is regular at that point. Takes a few years to actually achieve a regular ovarian cycle. So the first few years that you are responding to the HPG axis, right, ovulation is not a guarantee. Does that make sense? Could be happening, may not be happening, very inconsistent cycle to cycle. Takes about 2 to 3 years to achieve a regular ovarian cycle where you can regularly kind of determine what is the length of your ovarian cycle, and obviously then figure out when ovulation is possibly occurring, okay? So since you mentioned that, I'm going to go ahead and say this. Okay. So I said every ovarian cycle, right? We're choosing about 5 to 8 of these follicles, right? So at that point, we're going to call these follicles primary follicles, technically. Because, so these primary follicles are responding to FSH. They're going to become bigger, secondary follicles, so on and so forth, right? Getting ready for ovulation. But I'll tell you this, these follicles were actually selected not the month that they are used, they were selected about a year prior to the cycle in which they are used. So to your question, they're all primordial follicles. Then they get, let's say, 5 follicles get selected, right? These will start maturing into, what are they? Just becoming a little larger in size. Over the next year, they become those primary follicles. Then they enter the ovarian cycle, responding to the FSH and LH about a year later. Does that make sense to you? Right. Okay. Good. All right. So then here's the mature vesicular follicle. Again, with the primary oocyte, the zona pellucida, and your corona radiata. That's what's protecting the primary oocyte. So what happens? All right, back here, day 13. That day 13. About 24 hours prior to ovulation, right? So remember we said initially GnRH was upregulated, FSH and LH was released from the anterior pituitary. This allowed those predetermined follicles from like a year ago to basically get recruited into this ovarian cycle where they start responding to that follicle stimulating hormone and they start developing. Is that right? Okay, so then when we get to about 24 hours, I'm sorry, by day 5, we're shutting this down. Estrogen is guiding the rest of this development. But by day 13, increasing levels of estrogen is feeding information back to the hypothalamus. And for a very short amount of time, GnRH is active again. Follicle stimulating hormone and luteinizing hormone are released. But at that time, luteinizing hormone is the key player here. We call this LH surge. Is that right? So LH surge is going to act on this structure right here. You have your primary oocyte, isn't it? You know what? I think I have a better looking picture. But that primary oocyte is going to respond to the LH surge. Does what? We're going to complete meiosis. Well, let's write that. Primary oocyte. This is arrested in prophase 1, correct? Now in response to LH surge, what happens to this primary oocyte? We're going to complete meiosis 1, meaning we're going to come out of prophase 1, we're going to do metaphase 1, anaphase 1, telophase 1. We're going to wrap this up where one cell becomes 2 cells, correct? Except unequal division, we talked about this. You end up with what? One secondary oocyte, and then the other cell is the first polar body. That's what you end up with. Is that right? Both of these are going to start meiosis 2. This time we have the secondary oocyte arrested in metaphase 2. Right? So in this 24-hour period, all of these changes are occurring, all in response to LH surge. So yes, a vesicular follicle that is mature originally had that primary oocyte arrested in prophase 1. But 24 hours prior to ovulation, in response to LH surge, that primary oocyte completes meiosis 1, becoming 2 cells, unequal cells. All of that cytoplasmic content is here within the secondary oocyte. Talk to me about chromosomes, because you've completed meiosis 1. How many chromosomes do you have here? Is it 46? This was 46. 2n equals 46. What about the 2 daughter cells after meiosis 1? Haploid. It becomes haploid. So this is n equals 23. You see it? So, okay. So then what gets ovulated is this cell. Both, really. But this is what's getting ovulated. So we say very specifically, ovulation of the secondary oocyte. It would not be accurate if you said ovulation of the primary oocyte. Does that make sense? And it's not ovulation of the follicle either. Follicle is the entire package. It's not the entire package that is released from the ovary. Does that make sense to you? Right? The entire package is the vesicular follicle. What gets ovulated from the ovary is just the secondary oocyte, haploid now, because we've completed meiosis 1, plus that zona pellucida and the corona radiata. This entire thing here in the middle is what's ejected from the ovary into the uterine tube. Does that make sense to you? What remains, so the antrum, the rest of this vesicular follicle gets transformed into the corpus luteum. So that remains in the ovary, the corpus luteum. But of course, the secondary oocyte has been ejected and it's now in the uterine tube. Does that make sense to you? Yes, ma'am. I will go over that one more time. Yeah, yeah. Does that make sense so far? So what you're seeing here is the corpus luteum. This is the remnants of that vesicular follicle after ovulation has occurred, right? So obviously you don't have an oocyte in here anymore because that's already been ovulated. Okay. All right, let's keep going. I'll come back to her question as well. So follicular phase basically talks about development of the follicle, response to LH. We discussed all of that. Ovulation is literally an inflammation event. So this is where that secondary oocyte, as it's getting ready to be ovulated, it's going to it has to break through the ovarian wall. And so some pain is experienced in that process. So some females experience it and realize what's happening, and some don't. Okay, so it just kind of varies. Again, if you only have one dominant follicle, one dominant vesicular follicle, and only one secondary oocyte was released during ovulation, well, then that secondary oocyte has a possibility of being found by the sperm cell, penetration, and fertilization, resulting in one embryo, one zygote, right? What if you had two dominant follicles? Then two independent secondary oocytes would be ovulated, which could be fertilized by two different individual sperm cells, resulting in fraternal twins or triplets, and so on and so forth, right? I'll talk about identical twins a little later with respect to the next chapter, but that's really where you have one secondary oocyte fertilized by one sperm cell. You form this new individual, and then very, very early on in a process called cleavage, where you have rapid mitotic events, that's where that one embryo, right, zygote, splits into two. So now you have basically identical genetic content between both those siblings. Now you have identical twins. So I'll talk about that a little bit more in the next chapter. Corpus luteum, this is where the corpus luteum is formed. Yes, and the corpus luteum is going to start increasing something called progesterone. Okay, it's also going to maintain estrogen levels, but mostly progesterone. That's important. Oh, okay, all right then. There is no progesterone prior to ovulation. Progesterone is only after ovulation, because progesterone is made or secreted by that corpus luteum. Does that make sense to you? Okay, now we talked about this. Here you go, kind of showing you the different phases. These are all primordial follicles, primary follicles. You kind of see a secondary follicle right there. This is a very, very early vesicular follicle. Now you have more of a mature follicle here, kind of seen here as well. Again, you have a fluid-filled continuous antrum, and then you have initially your primary oocyte secluded, correct? And then in response to LH search becoming the secondary oocyte, it is surrounded by the zona pellucida and the corona radiata. It is this entire structure that gets ovulated, as you can see there, right? And then what's remaining of this structure gets transformed into the corpus luteum. The corpus luteum will continue to release progesterone and estrogen. It depends for how long. It depends on the fate of this secondary oocyte after it enters the uterine tube. Now, that secondary oocyte, we said, was viable for about 12 to 24 hours. So if penetration by sperm were to occur, it has to happen within 24 hours, give or take, right? Does that make sense to you? If that occurred, if penetration from sperm occurred, like that, yeah. So if this secondary, so this is the first scenario, if the secondary oocyte is actually penetrated by sperm, where you see fusion of the sperm's plasma membrane with the plasma membrane of the secondary oocyte. If that were to occur, right, and the contents of the sperm enter, I'm talking about the 23 chromosomes, as they start entering, this is when the secondary oocyte will, remember it was arrested in metaphase 1, it's now going to complete meiosis 2. That's only going to happen if penetration occurred with the sperm, right? Then the secondary oocyte becomes the ovum, and we form basically another polar body here. And this penetration of sperm is basically going to allow that first polar body to also complete meiosis 2, forming two more polar bodies. So the end result of this, again, if penetrated by sperm, is going to result in your 4 daughter cells. Ovum is the only one that we need to track, right? These 3 polar bodies, they're not useful for the whole development of a new individual. These will just disintegrate. That makes sense to you, right? Okay, what happens then if when the secondary oocyte is ovulated, it enters the uterine tube, but it does not encounter sperm. If it doesn't encounter sperm, it's going to make its way through the entire uterine tube, take about 4 or 5 days. It's going to enter the uterus, the body of the uterus, but it does not implant there at all. Therefore, this does not occur at all. This does not occur. We just end up with that secondary oocyte, first polar body, and then at the onset of menstruation, these two are released, right, in along with the menstrual blood, and as we break down the stratum functionalis layer of the uterus. So if no penetration with sperm occurs, this story ends here. Two daughter cells, it's a secondary oocyte and that polar body that is released during menstruation. But if penetration occurs with sperm in the uterine tube, then these two will divide one more time, right? I mean, we have our meiosis two occurring. Now you have your final four progeny, the daughter cells. Is that good? Okay, so if penetration occurred, we said we actually from the secondary oocyte becomes the ovum and a polar body and so on and so forth. The ovum is going to continue its journey into the uterus, where it's going to implant in the wall of the uterus, right, which is the endometrium layer. When implantation occurs, and really at that point, we don't call it the ovum. I'm gonna explain this, a lot of terminology. We call this implantation of blastocyst. So the ovum transforms into a few different things before it becomes the blastocyst, okay? But this is what's implanting. Where is it implanting? Into the endometrium. And the endometrium has nothing to do with the ovary, right? This is in the uterus. As it implants, okay, the earliest implantation event could be occurring about 6 to 7 days after ovulation. Because remember, the secondary oocyte is only viable for 24 hours. Penetration, if it were to happen, is probably happening by about day 15, right? And then it's going to continue its journey into the uterus, make its way another 4 or 5 days. And so the earliest implantation could be occurring about 7 days or so after ovulation. So if you were taking those early pregnancy kits, tests, right, very, very sensitive detection of HCG could be occurring as early as 7 days post ovulation, right? But really, as the implantation process proceeds and is successful, as the blastocyst makes its way firmly into the endometrium, what happens to HCG levels? Going to increase, correct? HCG is increasing within, it's coming from the blastocyst within the uterus, but this information is being fed back to the corpus luteum in the ovary, right? So I'm going to be safe. Let's say about day 24, which is 10 days or so after ovulation, right? The corpus luteum is asking, are HCG levels increasing? Am I seeing an increase in HCG? That's a question, right? Am I seeing that? Because if I'm seeing that, it means that not only penetration with sperm was successful, fertilization of the sperm's 23 chromosomes with the oocytes 23 chromosomes, that occurred, fertilization occurred, but then successfully that ovum, which then transformed into the blastocyst, was also implanted correctly within the endometrium. Not only fertilization, but also implantation is occurring. It's proceeding like it should. That's why you see HCG levels go up and stay that way. Because oftentimes, again, those of us that have been tracking cycles and looking for, a successful pregnancy, you may see, right, early pregnancy markers, HCG going up. And then as you continue to do these tests, those tests become, so initially you remember, or some of us at least will remember this, it's a very, very faint line, right? You got to look at it, the right light, like right angle. I'm like, oh, it's there. It's barely there. It's very faint, right? Then you wait 24 hours longer and you don't have to strain your eyes that much. You're like, oh, I see it. It's a positive, right? And then it becomes faster and faster and much stronger. Why? Because you're seeing increasing concentration of that HCG. How did that happen? Because implantation of the blastocyst is secure. Does that make sense? This is what the corpus luteum is looking for. This tells the corpus luteum we have a successful pregnancy, at which point I do not want to end this ovarian cycle. Because if you end the ovarian cycle, you will bring on menstruation and there's blood loss. You're tearing down tissue. You're tearing down what tissue? Endometrium, part of the endometrium, where you have this implanted blastocyst. So you'd be actually losing that blastocyst. Does that make sense? We don't want that. So if HCG levels are increasing, the corpus luteum is going to say, we got a successful pregnancy. We're not going to stop at day 28. We're going to keep going. We're going to keep going throughout the pregnancy. Does that make sense? Except the corpus luteum only stays alive. Remember, it produces progesterone and estrogen. Those are very important markers. We need those to sustain a viable, successful pregnancy. So corpus luteum will continue producing these two hormones for the first 2.5, 3 months or so during the pregnancy. While that's happening in the first 2.5 months, I'll talk to you about this, the placenta is developing. So the blastocyst is going to transform into several different things, but the placenta is forming back in the uterus, right? So at about 2.5 months, the placenta is ready to take over the role of the corpus luteum. So as soon as the placenta starts increasing or producing estrogen and progesterone, the corpus luteum is done with its job, right? At that point, it can start degenerating and transforming basically into scar tissue. Does that make sense to you? Okay, so let's add all of this information. Make sure we're tracking this. This is showing you post-ovulation, what's going on. So this is the follicle that we just saw ovulation from, right? First, it gets filled with blood. So it's like a blood clot, right? This is called the corpus hemorrhagicum. And then it becomes this structure called the corpus luteum, which is that densely packed structure filled with cells. It's this structure that is increasing estrogen and progesterone levels for the first 2.5 months or so of the pregnancy. But after the placenta takes over, the corpus luteum will start disintegrating, transforming into scar tissue, collagen. You see that? This is called the corpus albicans. This is the entire life cycle if you take it all the way through. But if not, again, if there's no pregnancy occurring, then the corpus luteum will just disintegrate, becoming scar tissue a lot faster then, right? I mean, okay. So let's talk about this whole hormonal regulation. I've already added a lot of information there, but I'm going to use your schematics to work my way through this. So throughout childhood, until you hit puberty, this axis is not active. So prior to birth, during fetal development, that axis was active to a certain extent because we need to develop female reproductive organs to a certain extent, right? But after birth, throughout childhood, GnRH is shut down, no FSH, no LH, right? Till you hit puberty. So really the trigger here, believe it or not, is actually from adipose tissue. The accumulation of fat, right? As you see widening of the hips and initial kind of development of breast tissue and all of that, it's accumulation of adipose tissue and a hormone that it produces called leptin that's going to signal to the HPG axis that it's time to stop these ovarian cycles, right, for the next few decades. Does that make sense to you? So then in response to leptin, as leptin levels increase, this axis becomes active. So you have GnRH being released by the hypothalamus, FSH, LH from the anterior pituitary that's going to target the ovary, and yes, you're going to start producing these hormones. Now, this is, like I said, this is not a very reliable pathway initially. It's going to take a few years to actually regulate this to where you have reliable ovarian cycles. That's why I said the first few years, it doesn't always have to be ovulatory, right? It hit or miss. It can kind of take that time. So when you have, it takes a little while to get this axis or this hormonal pattern regulated. When you see, when a female experiences their first menstrual period, this is called menarche, right? And then from there on out, right? I mean, all those ovarian cycles, again, takes a little while to actually establish it to become regular. And here's the whole HPG axis. Let's go through this. So I've got the hypothalamus up here, which is going to release GnRH. So I'm talking about every ovarian cycle. So let's imagine this is day 0 to this midpoint here, which is the ovulation time frame. So I'm discussing all the changes occurring during the follicular phase, right? So during the early follicular phase, the hypothalamus releases GnRH, which allows the anterior pituitary to release FSH and LH, correct? What does the FSH do? Do you see this follicle right here? This is like a maybe like a secondary follicle starting to become like, like early stages. The FSH is going to target these granulosa cells and basically prod it along to allow it to mature, right? Primary to secondary and so on and so forth. LH, on the other hand, is going to target this outer rim here. These are called the thecal cells. The thecal cells produce androgens, androgens like testosterone. Well, testosterone is not very useful to the ovary. So what I need to do is to take that androgen, move it inwards into the granulosa cells where you convert those androgens into estrogen. Do you see it? So this developing follicle is what's going to increase your estrogen levels. Do you see that? Right? So early on, this is what's happening. But then after about day 5 or so, you don't have FSH and LH anymore, right? Because you see these dotted lines here, this negative feedback. Inhibin, which we talked about even with spermatogenesis, same hormone. This is the hormone that's inhibiting the hypothalamus, the anterior pituitary, that entire axis. But estrogen continues to increase because you have a developing follicle, not just one developing follicle. Like I said, 5 to 8 developing follicles. Does that make sense to you? Now, as this proceeds in development, you now have your mature follicle here. This is your vesicular follicle. You have your primary oocyte arrested in prophase 1, zona pellucida, all of that. Here's your antrum, correct? Imagine that this is around day 13. What happens next, right? All right, what happens during day 13? This is where you have temporary override of that HPG axis, right? GnRH is released one more time. LH is the prominent player here. Now, what's the effects of LH? All right, this is what happens. LH is going to target the primary oocyte, allow it to complete meiosis 1, becoming the secondary oocyte in the primary, the first polar body. The secondary oocyte is what you now see within that vesicular follicle. It is arrested in metaphase 2, right? Then ovulation occurs, right? Ovulation is where that secondary oocyte is ejected out of the ovary into the uterine tube. And then after ovulation, the rest of the contents or the remnants of the vesicular follicle gets transformed into the corpus luteum. We know what the corpus luteum did. It increased progesterone levels and estrogen. Okay, so let's show you the rest of this. So we explained the first part. Now let's talk about as we are approaching ovulation. As we're approaching ovulation, increasing estrogen levels is going to feed back to the hypothalamus. So temporarily, for a couple of days, right? GnRH is released again. You see FSH, but really LH is the prominent player here. What does the LH do? Like I said, that primary oocyte is going to get transformed to the secondary oocyte. It allows for the ovulation to occur, right? And then the rest of this structure gets transformed into the corpus luteum. Once the corpus luteum is formed, we're going to start increasing, we're going to keep those estrogen levels high, but at the same time start increasing progesterone, but also corpus luteum increases inhibin levels again. And inhibin does what? Goes back to the hypothalamus, shuts down the GnRH pathway. Because if this LH continues, what's going to happen? The rest of these follicles that were not quite dominant at day 14, well, they're going to become dominant. And then you will have multiple ovulation events. Does that make sense? We're gonna curb all of this. Everything is regulated, right? Does that make sense to you? So increasing estrogen and progesterone levels tells the and also HCG levels tells the corpus luteum that it needs to stay intact, right? But what happens when HCG is not found? Means pregnancy did not occur, fertilization did not occur. So what happens to the corpus luteum? It's going to start breaking down, right? Disintegrating. As the corpus luteum disintegrates, tell me what do you think is going to happen to progesterone and estrogen? It's going to start dropping. Dropping levels of progesterone and estrogen shuts down this cycle and lets you know it's time to start all over again and brings on menstruation. That makes sense? Yeah? Okay. This entire time frame, right, from the time ovulation occurs to what's the fate of the oocyte, the secondary oocyte, did it get fertilized by sperm, right, or did it not? Are we looking for HCG around this time frame? If HCG levels are not high enough, then we're going to abort this cycle. This is very constant because there's a specific time that it takes for each one of these steps to occur. And it happens in the same kind of sequence, same duration. There's no variation there. Does that make sense? Okay. So let's keep going. We already talked about what happens if there's no fertilization. This is kind of showing you all the different hormones pre and post ovulation. So, okay, ovulation is right here in the middle. Everything in blue is prior to ovulation. Everything in green is after ovulation, right? So before ovulation occurred, during the first few days, you're seeing FSH in red and LH in blue, right? You're going to see a slight increase there. That's allowing, okay, you see all these primordial follicles here? We've got a few follicles that are selected responding to the FSH and LH. They're going to start maturing. You see that? Correct? As they mature, what happens? Estrogen levels go up. That's my orange marker right there. You're going to start seeing estrogen levels go up, correct? But remember, all these levels of FSH and LH, they drop for the rest of this ovulation, pre-ovulation period. As ovulation occurs, ovulation of the secondary oocyte occurs in response to this LH surge, then what happens? Secondary oocyte is ovulated. The rest of the structure becomes the corpus luteum. Now watch. What does the corpus luteum do? Look at the orange tracing. That's estrogen, right? You're going to keep those estrogen levels fairly constant. But what's the green tracing? Progesterone. You see, progesterone was flat prior to ovulation. Nothing secreted that progesterone. You see progesterone levels starting to increase. This is after ovulation in response to that corpus luteum. Make sense? What happens to FSH and LH after ovulation? Don't need it, right? So it's going to decrease until it's needed again at the start of the next cycle. Do y'all see that? Okay, so this is the ovarian cycle. What I'm going to map out next is the uterine cycle. As these changes are occurring in the ovary, I also want to describe to you what are the changes occurring in the uterus corresponding to these same time frames, right? So let's do this. Okay, I'm going to go right there. This is the uterine cycle. And we're also going to do day 5. So 0, 5, 14, 28. Okay, so days 1 to 5 is what I call the menstrual phase. The rest of it leading up to ovulation is the proliferative phase, and then the last part is the secretory phase. So this part, this initial first 4 or 5 days, menstrual. Then the rest of this until ovulation, proliferative. And then everything else after ovulation, secretory. So if you're looking at this, right, okay, the ovarian cycle from start to ovulation is called the follicular phase. So the follicular phase corresponds time-wise to both the menstrual and the proliferative phase within the uterus. Do you see that, right? Follicular phase equals menstrual plus proliferative. Same time frame. After ovulation, we're calling this the luteal phase in the ovary, which corresponds to the secretory phase in the uterus. Do you see that? All right, so let's talk about this. So when you're looking at the wall of the uterus, there are 3 layers there, starting with what's facing the lumen on the inside is the endometrium. Then you have the bulk of the uterine wall, which is smooth muscle, that's myometrium. That's very important for labor, right? We'll cover that next class. And then the outermost is the perimetrium. So what I'm interested in right now is the endometrium, because the endometrium has 2 layers, or 2 regions rather. So the layers that are closest to the lumen is called the stratum functionalis. Where is it? Yep, okay, it'll come up later. And then the layers a little further away are called the stratum basalis. So here's what's happening. During the menstrual phase, specifically During here, what's happening? So why would you be bringing on menstruation? Well, if the corpus luteum in the ovary in the previous cycle did not detect HCG levels, it's going to disintegrate. Progesterone levels are going to drop. Estrogen is going to drop. Dropping levels of progesterone and estrogen ends this cycle and brings on the menstrual phase within the uterus. Is that right? Okay. So what I'm doing here is blood vessels are being broken, right? Obviously blood loss, but also tissue loss. So what tissue loss? Within the wall of the within the wall of the uterus, I talked about the endometrium, specifically the layers of the stratum functionalis. Stratum functionalis layers are shed. That's what's lost. Does that make sense? Right? Okay, now in the body of the uterus, you had a secondary oocyte, and that first polar body that never, I mean, that secondary oocyte was never penetrated by sperm. Well, that's also lost as part of that menstrual process. Okay, so then in this next few days here, leading up to ovulation, this is the proliferative phase. This is when the layers above the stratum functionalis, which is the stratum basalis, this consists of stem cells. They're going to undergo rapid mitosis, where what? One parent cell becomes two daughter cells, becoming 4, 8, so on and so forth. So the stratum basalis is going to rebuild the functionalis layer that you just lost during the menstrual phase. So stratum basalis, it rebuilds functionalis layers. That makes sense to you? This is all again in preparation for what if ovulation occurred and that secondary oocyte was penetrated by sperm. Then you want a hospitable environment, right? You want a healthy endometrium for that implantation to occur to sustain a pregnancy for the next 9.5 months. Is that right? So we got to rebuild in preparation for what if, what if we have a viable pregnancy. Does that make sense? And I'm going to just say this, the last part, the secretory part, which is after ovulation, is really, okay, we know that ovulation has occurred. So now we're getting ready for, in case it was fertilized, then we want to sustain that implantation. So this is really where you are, let's say, increased blood vessels. So this is all prep work, okay? Increased blood vessels, meaning more blood flow, nutrients, all of that. You have your endometrial glands that are going to start releasing secretions, like I'm talking nutrients, right? All of this. This is all prep work for implantation. I'm going to say for possible implantation. Does that make sense? All right, so let's add this information. So the uterine cycle is not that complicated. So here's the menstrual phase. Like I said, dropping levels of estrogen and progesterone is going to signal that the stratum functionalis layer needs to be shed. So this could last about 5 days. Proliferative phase is when you have estrogen levels that are rising, correct, in the ovary. That's going to signal, okay, it's time to rebuild the stratum functionalis layer. And also, remember, progesterone is actually secreted after ovulation. But those receptors for progesterone in the wall of the uterus have to be kind of upregulated during this proliferative phase because they need to be able to respond to increasing levels of progesterone to sustain a pregnancy should it occur. So it's again kind of anticipation, right? Oh, one more thing. Cervical mucus. So cervical mucus is thick or thicker till we approach ovulation. So closer to ovulation, it starts to thin out to allow better penetration of the sperm so that it can make its way into the uterine tube, easier passage, basically, right? So that's definitely happening too. And the last part here, the secretory phase, again, I said, the functionalis layer is getting ready for implantation, should it happen. So it becomes more secretory in nature. That's why it's called the secretory phase. Endometrial glands will start producing nutrients, more blood flow, getting ready, okay? If fertilization does not occur, well, all of this work was not needed, right? If fertilization does not occur, we're going to, as the corpus luteum is disintegrating, this is going to end the uterine cycle and bring on menstruation again. Yes, ma'am.
So if there was a miscarriage, how long would it take for the miscarriage?
To what start again? The entire ovarian cycle? Oh, that depends. That depends female to female. So hormonal regulation, the balance has to be, I mean, it could be the very next month, or sometimes there might be gaps because we're needing to kind of like, again, depends on also, right? I mean, what type of miscarriage? Is it hormonal regulated? Is it structural related to the endometrium not being able to support that pregnancy? So there's a lot of things that would need to be rebuilt or reach homeostasis again before you can kickstart the next ovarian cycle. So that's a interesting question, but it cannot be answered very easily because it depends on case by case scenarios, right? Could be very next month, could take a few months, sometimes could result in other complications where you have to intervene, could take a year. It just kind of varies, okay? But I guess the easiest answer is could be as early as next month, right? Okay. So let's talk about the uterine cycle then here. On the bottom, we talked about the top three, which is all ovarian. Here are the changes occurring in the uterine cycle. If you're just looking at the endometrial layers, the basal layer is this darker portion on the bottom, and the functionalis layer is the lighter portion on the top. So during the menstrual phase, notice how you're shedding, you're getting rid of the functionalis layers. During the proliferative phase, it's the basalis layer that's recreating or rebuilding the functionalis layers. You also start to see more blood flow here, more arteries, right? And then in the secretory phase, a lot of arteries. So bringing more blood flow, bringing more nutrients, oxygen, endometrial glands that are producing those nutrients as well. So all of this is in preparation for a successful pregnancy should implantation occur. How do you make that pregnancy thrive, right? So that's all preparation. But as the corpus luteum disintegrates back in the ovary, what happens to the stratum functionalis layer in the uterus? It starts to get shed, right? I mean, and that brings on menstruation. Okay. All right, last few things. What are the effects of estrogen? We've already talked about it, right? I mean, estrogen plays a huge role in oogenesis. At the time of puberty, also inducing secondary sex characteristics, okay? Progesterone, okay, you know how progesterone plays a big role here in the uterus, all again in preparation for implantation to occur. But should pregnancy occur, how do you sustain the pregnancy? Progesterone is the marker that's super important, along with other things. But progesterone is that hormone that needs to be pretty stable throughout the pregnancy. In fact, as you get closer to labor and delivery and start experiencing Braxton Hicks, false labor, all of that, it's progesterone levels starting to fluctuate, starting to drop a little. That's kind of actually signaling to mom, and it's all coming from the fetus as well, that it's time for baby to meet world, right? I mean, so that's kind of all kind of tied into the onset of labor. But throughout the pregnancy, we want progesterone levels to stay high. This is going to be important towards the end of the pregnancy, helping breast tissue get ready for lactation, right? Things like that as well. I think that wraps it up. Genetic sex, I think we're good with this part, okay? Um, Puberty, I've already talked about this. Menopause. So when menstruation ends and you do not have a menstrual period for at least a year, that's when it's safe to say you've entered menopause, right? At that point, you do not have an ovarian cycle, you do not have ovulation events and all of that. So, of course, fertility is low, right? At that point, things can happen though. All right, so I'm going to stop right there and end this. How are we doing? There's a lot of hormonal regulation with the female reproductive system. So just kind of take a look at that and see if we need to go over something next class. But I'll go ahead and get started with this next chapter to define a few terms and just kind of get started, not do a whole lot though. All right, so let's see. Remote users, I'm sharing my next set of slides. This is pregnancy and fetal development. Can you see it? Okay, perfect. All right, let's go ahead and define a few things. All right, so this is all in continuation what we've been discussing, right? So ovulation occurs, the secondary oocyte that is arrested in metaphase 2 is ejected into the uterine tube. It is viable for what? 12 to 24 hours, during which time penetration by the sperm could occur. And if that occurs, then we will define a process called fertilization. They're almost synonymous. Like as soon as penetration occurs, fertilization will occur. So that it can be used synonymously, but let's separate it out. Penetration is where you see the fusion of the sperm plasma membrane with the fusion of with the oocytes plasma membrane. But then the actual genetic content as they merge together, 23 from sperm plus 23 from the oocyte, as they merge together and become one new cell, that's what I call fertilization. All right, so let's talk about that. The developing offspring is called the conceptus. It has different terms to it. So let's define this. Gestation period is basically from the time of your last menstrual period until birth. So technically, you are not pregnant during the first 2 weeks of your gestation period, right? I mean, like this is when you're preparing for ovulation. There is no possibility that sperm has already encountered anything here. This is prior to ovulation. But those first 2 weeks are included in the gestation period clinically because not every female is tracking and charting and knows exactly when your ovulation event occurred and if you were timing intercourse or whatever, or artificial insemination, whatever the case may be. Not everybody is tracking that, but most everybody, females, would know when was the start of your last period, right? So that's why it's included in that gestation period. But remember, technically, those first 2 weeks, you're not pregnant, right? Okay. Um, we call the developing conceptus from the time of fertilization all the way through basically the first 8 weeks, the first 2 months, right? We call this the embryo. 9 weeks onwards until birth, we call this fetus. So next class, we will talk about changes occurring in the embryo and fetal development changes as well. For right now, I'm just kind of getting us started with this discussion. We said the oocyte, the secondary oocyte that's ovulated is viable for up to about 24 hours. This is when the sperm would need to penetrate that oocyte. Remember, the sperm, though, is viable for a lot longer, for up to 48, maybe 72 hours as well, right? Just kind of depends on the viability of that sperm. It varies. So fertilization is really when the chromosomes of the sperm fuse with the chromosomes of the secondary oocyte. So let's go ahead and explain this. How do you accomplish fertilization? So word slides explaining different things here. I'm going to go ahead and talk about all of these events using this schematic. All right. So the first thing I want to talk about is, let's see. We'll be tracking a few things here, and I'll probably rebuild all of this next class as well. So the first thing that you see here is here's the secondary oocyte that's been ovulated. Remember, this has been ovulated. So it's ovulated, it's arrested in metaphase 2. It is surrounded by this gray layer, which is the zona pellucida, and a few layers of the corona radiata, right? That's what was ejected out of the ovary, and now it is in the uterine tube. Does that make sense? The first thing is, of course, approach from all of those sperm cells, right? And what I'm going to explain to you is even prior to the approach. Let's talk about capacitation. When sperm is deposited into the vaginal canal, all right, by gravity, a lot of it could leak out, right? Others are going to make their way into the uterus where they're going to encounter the natural macrophages and other immune cells that are resident to the female body that encounter the sperm saying that's foreign and of course mount an immune response against it. But if you recall, semen consists of sperm, but also all the other fluids, right? The secretions related to it. And one of the things there are immune suppressants. So that's going to be a natural kind of component there that's going to suppress, not completely eliminate, but suppress the effect of the female reproductive system, right? Okay, really only 100, maybe a few thousand of those sperm cells compared to millions that are deposited, right, will actually make its way past the vaginal canal, past the cervix, remember the external os, internal os, and enter the body, the lumen of the uterus. So before it makes its way, right, Sperm cells are going to go through a process called capacitation. Let me back up here for a second. Capacitation is where the sperm is preparing for that upward journey. It's against gravity, right? It's reverse peristalsis, really. So it has to make its way through the lumen of the uterus, make its way in the uterine tube, and get to almost the end, the distal portion of the uterus, because it needs to be ready, waiting for that ovulated oocyte, correct? So, got prepared for the journey, first thing. ATP reserves have to be built up. That's all part of this capacitation process. So for that preparation, this is where, remember we said clotting factors are also part of the semen, right? The clotting factors help to make that fibrin mesh, which allows those sperm cells to cling to the vaginal wall for just a little bit, right? Allowing the sperm cells to regroup, utilize fructose and other like citric acid and things like that. That's part of semen. Utilize that to generate ATP because we got to swim upstream, right? So that's a lot. That's a bit of a journey. Lots of reserves that are required for that. And then remember how the sperm had three different regions. The head region, which has the 23 chromosomes inside of it, protected by an acrosome, like a helmet region. Well, we're going to make that acrosomal, that helmet region, a little bit more fragile, okay? Because as we start encountering ultimately that oocyte, I need to be able to release the contents of the head region, right? So the acrosome basically starts becoming a little bit more fragile. And then all of this is part of that capacitation process, right? It could take, I don't know, a few minutes, could take up to an hour, sometimes few hours. All of that has to happen. That's the capacitation process. Once that's occurred, then you see how those sperm cells are approaching, hopefully, that secondary oocyte, okay? As it makes its journey through the vaginal canal, there's plenty of other cells there, right? I mean, we're talking the endometrial layer, immune cells, lots of other things happening. How does the sperm know not to bind a macrophage or an epithelial cell or whatever, right? Y'all remember in AMP1, we talked about something called the glycocalyx. Every cell membrane, you had the phospholipids, but you had like a carbohydrate sugar coating. It was a signature pattern that identifies each cell. The sperm is looking for that specific glycocalyx signature pattern on the secondary oocyte. It needs to recognize that and knows, here's my target. That's the cell I need to bind. I don't need to be binding any one of these other macrophages, dendritic cells, epithelial cells, or anything else because the glycocalyx is unique to every cell. Does that make sense? Okay. As it approaches the secondary oocyte, here's what's going to happen. This is showing you the secondary oocyte a little bit better, much more zoomed in. This is the interior of the secondary oocyte. Here's the plasma membrane of the secondary oocyte, the zona pellucida, and those are those layers of corona radiata. You see that? As the sperm makes its way, it's easy to get the corona radiata out of the way. It's just cells, right? Move it out of the way. Here's what happens next. The sperm is then going to bind receptors on the zona pellucida. Does that make sense? Right? This is what I call binding, right? And this is going to trigger something called the acrosomal reaction. When the sperm binds receptors on the zona pellucida, inside the sperm, calcium levels are going to rise. Okay. What it does then is, remember that acrosome that was a little bit brittle, fragile, right? It's going to basically break, releasing hydrolytic enzymes. Does that make sense? Those hydrolytic enzymes are able to punch holes through the zona pellucida, right? Allowing the sperm to go further inwards, right? So kind of approach the plasma membrane of that oocyte. So that's what I call the acrosomal reaction. I have to breach the zona pellucida. Okay, acrosomal reaction. Once the sperm reaches the oocyte's plasma membrane, this is where binding will occur. So this is what I mean by penetration, right? Penetration of the sperm's plasma membrane with the oocyte's plasma membrane. Does it make sense? This is going to allow for the next thing to happen, which is flat to polyspermy. And as this is happening, inside the secondary oocyte, right? This binding is what going to allow the secondary oocyte to come out of metaphase 2 and then go into what? Anaphase 2 and telophase 2, allowing you to form the ovum and all of this to occur. Makes sense to you? So that penetration, binding of the oocyte and the sperm's plasma membranes will allow the ovum to be created on the inside. But on the outside, what's happening? So there are, when you see binding occurring here, calcium within the oocyte will go up, which is going to allow these granules here, these are called cortical granules. It allows these cortical granules to undergo exocytosis. And as a result, what happens then is that zona pellucida is a very kind of a flexible, kind of a gelatinous kind of a membrane, but the cortical granules are going to change the consistency of that zona pellucida, hardening it, right? So of course, you kind of see just a few sperm cells. Imagine hundreds of sperm cells, right? All right there at the zona pellucida. One lucky sperm cell is going to make its way all the way through that zona pellucida. As soon as one sperm binds the plasma membrane of the oocyte, we want to block the other sperm from making its way into it, into the secondary oocyte, right? How do you block it? By changing the consistency of the zona pellucida, where you've got the other sperm cells, what, nanoseconds away from that original sperm cell, right? And we're basically gonna, because you're changing it, those other sperm cells get clipped off. They're not able to gain entry into the oocyte. Does that make sense? You don't want multiple sperm cells entering that oocyte. The oocyte, which now is the ovum, right, has 23 chromosomes. The sperm also has 23 chromosomes. You only want 23 to merge with 23. So you have that first cell, the zygote. Does that make sense? If multiple sperm enters, then you don't have just 23 plus 23. You have 23 times God knows how many. Does that make sense? That is no good. We want to block polyspermy. Single sperm needs to fertilize or needs to penetrate and then fertilize the secondary oocyte. Okay, let's keep going. So we talked about block to polyspermy. So let's talk about fertilization. So as soon as this occurs, right, here's what happens. Now, let me back up for a second, showing you. Once you're blocking, once you change the consistency of the zona pellucida, this one cell, this one sperm is going to release the nucleus that contains the 23 chromosomes. The rest of this, the flagellum, is really not needed. I mean, its role has been accomplished, right? It's just we're gonna just release the nucleus with the 23 chromosomes into the secondary oocyte, which now I call the ovum, right? The ovum has its 23 chromosomes. The sperm nucleus has its 23 chromosomes. So now what we're going to explain is this is what's going to happen. The sperm nucleus is going to swell. We call this the male pronucleus. The ovum's nucleus is going to swell, female pronucleus, right? So you got these two nuclei, male sperm versus the ovum right there. Remember that each nucleus has its own nuclear membrane, right? So what I need to do is allow for the nuclear membranes to be broken. That allows the 23 chromosomes from the male, the sperm, to actually fuse now with the 23 chromosomes of the ovum. When fusion occurs, we call this fertilization. You see that? Penetration first needs to occur that allows this process to complete, allows block to polyspermy. But then immediately after that, you actually see N equals 23 plus N equals 23 coming together. Fusion of those chromosomes, where now you have a diploid zygote being formed. Make sense? Okay, that's kind of what's shown there. So I'll go ahead and talk about the rest next class. This basically maps out the once the fertilization event occurs. So now we call this what? A fertilized ovum. You can call it a zygote, right? And then the name's going to change as the rest, as this fertilized ovum continues its journey through the uterine tube. So y'all should have covered this in lab. Y'all should have looked at fertilization models through the uterine tube. Do you remember this? You'll actually have a model looks very similar to this, all the changes that need to occur. And then I'll tell you about the blastocyst forming and so on and so forth. So there is some information to cover on Thursday. Please do come somewhat prepared so that we can go through this rather quick. Again, genetics, has already been covered. I'm only going to cover certain topics, probably 5 to 7 minutes. That's it. Okay. Yes, ma'am. Does somebody have a question? Yes. If it's triplets, we're talking about three secondary oocytes that were ovulated, penetrated by three individual sperm cells. Yeah. Okay. I'm going to stop sharing remote users. I'm going to say bye to you all. Have a wonderful rest of your day.